So hello everyone, welcome back to my channel. Today we are going to study Unit 7th, that is Genetics and Evolution. The work of Mendel and others who followed him gave us an idea of inheritance patterns. However, the nature of those factors which determine the phenotype was not very clear, as these factors represent the genetic basis of inheritance. Understanding the structure of genetic material and the structural basis of genotype and phenotype conversion became the focus of attention in biology for the next century. The entire body of molecular biology was a consequent development with major contribution for Watson, Crick, Nirenberg, Khorana, Korenberg's father and son, Benzer, Monod, Brenner, etc. Parallel problem being tackled was the mechanism of evolution. Awareness in the areas of molecular genetics, structural biology, and bioinformatics have enriched our understanding of the molecular basis of evolution. In this unit, in the structure and function of DNA and the story and theory of evolution have been examined and explained. James Davy Watson was born in Chicago on 6 April 1928. In 1947, received BSc degree in zoology. During these years, his interest in bird watching had matured into a serious desire to learn genetics. This became possible when he received a fellowship for graduate study in zoology at Indiana University, Bloomington, where he received his PhD degree in 1950 on a study of effect of hard X-ray on the bacteriophage multiplication. He met Crick and discovered their common interest in solving DNA structure. Their first serious effort was unsatisfactory. Their second effort, based upon more experimental evidence and better appreciation as the nucleic acid literature, resulted in March 1950 in the proposal of complementary double helical configuration. Francis Harry Crumpton Crick was born on 8 June 1916 at Northampton, England. He studied physics at University College London and obtained a BSc in 1937. He completed PhD in 1950 on a thesis in light X-ray diffraction, polypeptides and proteins. A critical influence in Crick's career was his friendship with J.D. Watson, then a young man of 23, leading to 1953 proposal of double helical structure of DNA replication. Crick was made on FRS in 1959. The honors of Watson with Crick include the John Collins Warren Prize of Massachusetts General Hospital in 1959, the Lasker Award in 1960, Research Corporation Prize in 1969, above all, the Nobel Prize in 1962. Chapter 4, Principles of Inheritance and Variation. Have you ever wondered why an elephant always gives birth only to a baby elephant or some other animal, or why a mango seed forms only only a mango plant and not by any other plant? Given that they do are the offspring identical to their parents, or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other, or sometimes even so different? These are several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parent to offspring. Inheritance is the process by which characters are passed on from parents to progeny. It is the basis of heredity. Variation is the degree by which progeny differs from their parents. Human knew from as early as 8,000 10,000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variation that were naturally present in the wild population of plants and animals to selectively breed and select for organisms that possess desirable character. For example, through artificial selection and domestication from ancestral wild cows. We have well-known Indian breeds in Sahiwal cows in Punjab. We must however recognize that through our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of these phenomena. Mendel's law of inheritance. It was during the mid 19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiment on garden peas for 7 years and proposed the law of inheritance in living organisms. During Mendel's investigations into inheritance pattern, it was for the first time that statistical analysis and mathematical logic were applied to the problems in biology. His experiment had a large sampling size, which gave a greater credibility to the data that he collected. Also, the confirmation of his inferences from experiment on successive generation of his test plan proved that his result pointed to general rules of inheritance rather than being unsustained ideas. Mendel investigated character in the garden pea plant that were manifested as two opposing traits in, for example, tall or dwarf plant, a yellow or green seed. This allowed him to set up a basis framework for rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observation and the complexity inherent in them. Mendel conducted such artificial pollination or cross pollination experiments using several true breeding pea lines. A true breeding line is one that having undergone continuous self pollination, so the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding plant varieties, a pair of which were similar except for one character of contrasting traits. traits. Some of the contrasting traits selected were smooth or wrinkled seed, yellow or green seed, inflated or constricted green or yellow pods, and tall or dwarf plants. Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plants to study the inheritance of one gene. He collected the seed produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation, also called filial progeny or F1, Mendel observed that all the F1 progeny plants were tall, like one of its parent, none were dwarf. He made similar observation for the other pairs of traits. He found that the F1 always resembled either one of the parent, and that trait of the other parent was not seen in them. Mendel then pollinated the tall F1 plant, and his surprise found that in filial 2 generation, some of the offspring were dwarf. The characters that was not seen in F1 generation was now expressed. The proportion of plants that were dwarf were 1/4 of the F2 plant, while 3/4 of the F2 plants were tall. Tall and dwarf traits were identical to their parental type and did not show any blending. That is, all the offspring were either tall or dwarf, none were in between height. Similar results were obtained with the other trait that he studied. Only one of the parental traits was expressed in F1 generation, while at F2 stage both the traits were expressed in the proportion 3:1. The contrasting trait did not show any blending at either F1 or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down unchanged from parent to offspring through the gametes or successive generation. He called these things as factors. Now we call them as genes. Genes, therefore, are the unit of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. Alleles. They are slightly different forms of the same gene. If we use alphabetical symbols of each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of height, T is used for tall trait and small t for dwarf, and T and T are alleles of each other. And in plant, a pair of allele for work with TT or small this TT or this TT. Mendel also proposed that in a true breeding tall or dwarf P variety are the allelic pair of gene for a height are identical or homozygous. TT and TT respectively. TT and TT are called genotype of the plant, while the descriptive term tall and dwarf are phenotype. What then would the phenotype of plant that had phenotype TT? As Mendel found the phenotype of F1 heterozygote TT to be exactly like the TT plant in appearance, he proposed that in a pair of dissimilar factors, one dominate the other as F1 and hence is called dominant factor, while the other factor recessive. In the case T is dominant over small t, that is recessive. He observed identical behavior for all the characters, traits, pairs that he studied. It is convenient to use a capital of lowercase of an alphabetical symbol to remember this concept of dominance and recessiveness. Do not use T for tall and D for dwarf, because you will find it difficult to remember whether T and D are alleles of the same gene character or not. Alleles can be similar as in case of homozygote. T or TT can be dissimilar in the case of heterozygote. Since the tea plant is heterozygous, controlling character, it is monohybrid, and the TT is an monohybrid cross. The observation that the recessive parental is expressed without any blending in the F2 generation, we can infer that when tall and dwarf plants produce gametes by the process of meiosis, alleles of parental pair separate or segregate from each other, and only one allele is transferred to a gamete. This segregation of alleles is some process, and so there is 50% chance of gamete containing either allele or as have been verified by the result of crossing. In this way, the gametes of the tall TT plant have the allele T and the gamete of dwarf TT plant have the allele T. During fertilization, the two alleles T from one parent through the pollen and T from the parent again through the egg are united to produce zygotes that have one T allele and one T allele. In other words, the hybrids have TT since these hybrids contain alleles which express contrasting traits. The plant are heterozygous. The producing gametes by parent, the formation of zygote, the F1 and F2 plant can be understood by from a diagram called Punnett square as shown in figure 4.4. This is Punnett square. These are Punnett squares. It was developed by British genetics Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotype of offspring in genetic graph. The possible gametes are written on two sides, usually the top row and left column. All possible combinations are represented in box below in the square, which generates a square output from form. The Punnett square shows the parental tall TT and dwarf TT female plant. The gametes produced by them F1 TT progeny, F2 plant is genotype TT are self-unrelated. The symbols this and this are used to denote female and male of the F1 generation respectively. The F1 plant of genotype TT when self-pollinated produce gametes of the T genotype T and T in equal proportion when fertilization takes place. The pollen grains of genotype T have 50% of chance to pollinate X of the genotype T as well as genotype T. Also, pollen grains of genotype T have 50% chance of pollinated X of genotype T as well as of genotype T. As a result of random fertilization, the zygote can be of genotype TT, TT, or TT. From the Punnett square, it is easily seen that 1/4 of random fertilization led to TT, 1/2 led to TT, and 1/4 to TT. So, their F1 have genotype TT, but the phenotypic character seen is tall at F2, 3/4 of plant are tall, whereas some of them TT are other TT. Externally, it is not possible to distinguish between the plant with the genotype TT and TT. Hence, within the genotype pair TT, only one character T crawl is expressed. Hence, the character T or tall is said to be dominate over the other allele. T or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall, so the genotype is TT, and in the F2, 3/4 of the plants are tall. So genotypically 1/2 are TT or 1/4 are TT. This led to phenotypic ratio of 3/4 tall, 1/4 TT plus 1/2 TT and 1/4 TT. It means a 3:1 ratio 1, but a genotypic ratio of 1:2:1. The 1/4 ratio, 1/2 ratio, 1/4 of TT is a mathematical condensable to the form of binomial expression AX plus BY square that has the gametes bearing genes T or T in equal frequency of 1/2. The expression is represented as given below. 1/T plus 1/T का whole square equal 1/2T plus 1/2T x into 1/2T plus 1/2T, that is expanded its whole square written here. So equal 1/4TT plus 1/2TT plus 1/4TT. As you know, the whole square is a plus b whole square is equal to a square plus b square plus 2ab. The same is here. Mendel self-pollinated the F2 plant and found that dwarf F2 plants continue to generate dwarf plant in F3 and F4 generation. He concluded that the genotype of dwarf was homozygous TT. What do you think he would have got had he self-pollinated to a tall F2 plant? From the preceding paragraphs, it is clear that so the genotypic ratios can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait. It is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has TT or T2 composition cannot be predicted. Therefore, to determine the genotype of tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This is called a test cross. In a typical test cross, an organism pre-plants here showing a dominant phenotype and whose genotype is to be determined is crossed with the A recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of test organism. Figure 4 shows the result of typical test crossover where violet color flower V is dominant over white colors flower. Using Punnett square, try to find out the nature of offspring or test require. What ratio did you get? Based on his observation on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called principles of law of inheritance. The first law, or law of dominance, and the second law of segregation. Law of dominance. First, characters are controlled by discrete units called factors. Second, factors occur in pairs. Third, in a dissimilar pairs of factors, one member of the pair dominates the other. The law of dominance is used to explain the expression of only one of the parental character in monohybrid cross in the F1 and the expression of both in F2. It also explains the proportion of 3:1 obtained at the F2. Law of segregation. This law is based on the fact that allele do not show any blending and that both the characters are recovered as such in F2 generation. So one of these seen at the F1 stays through the parent content to allele during gamete formation. The factor or allele of a pair segregate from each other that the gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar while a heterozygous one produces two kind of gamete each having one allele with equal proportion. Incomplete dominance. When experiment on peas were repeated using other traits in other plant, it was found that sometimes the F1 had phenotype that did not resemble either of the two. Parent was in between the two. The inheritance of flower color in the dark flower. Snapdragon or snapdragon or antirrhinum is a good example to understand incomplete dominance in a cross between true breeding red flowers and true breeding white flower plant. F1 was pink. When the F1 was self-pollinated, that resulted in the ratio red to ratio pink, ratio 1 white. Here the genotype ratios were exactly as we would accept in any other Mendelian monohybrid cross, but the phenotype ratios are changed from the ratio 1 dominant to ratio recessive ratio. What happened was that R was not completely dominant over R, and this made it is possible to distinguish RR as pink and from RR and RR mean red and white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand that a gene does what a gene does. Every gene, as you know by now, contains the information of to express a particular trait in a diploid organism. There are two copies of each other gene. It means as a pair of alleles. Now, these two alleles need not always be identical, as in heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on and in the next chapters, which modify the information that particular allele contain. Let us take an example of a gene that contain the information of producing an enzyme. Now there are two copies of this gene, two allele forms. Let us assume, as is more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate, as theoretically the modified allele could be responsible for production of the normal or less efficient enzyme or non-functional enzyme or no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele. It means it will produce the same phenotype or trait, resulting in the transformation of substrate as such. Equivalent allele pairs are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype or trait will be only be dependent on the functioning of the unmodified allele. Unmodified allele, which represent the origin phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Codominance. Till now, we were discussing crosses where F1 resembled either of the two parents or was in between. But in the case of codominance, the F1 generation resembles both the parent. A good example is different type of red blood cell that determine ABO blood grouping in human being. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cell has sugar polymer that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has three alleles IA, IB, and I. The alleles IA and IB produce a slightly different from the sugar, while allele for I does not produce any sugar because humans are diploid organisms. Each person possesses of any two of three I gene alleles in IB are completely dominant over I. In other words, when IA and I are present, only IA expresses because I does not produce any sugar, and when IB and I are present, IB expresses. But when IA and IB are present together, they both express their own types of sugars. This is because of codominance. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, there are six different combinations of these three alleles that are possible, and therefore a total of six different genotypes of human ABO blood types. How many phenotypes are possible? Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here we can see that there are more than two, it means that three alleles governing the same character. Since in an individual only two alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may be produced more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has two alleles, B and B. Starch is synthesized effectively by BB homozygotes, and therefore large starch grains are produced in contrast. BB homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of seeds, BB seeds are round and BB seeds are wrinkled. Heterozygotes produce round seeds, and B seems to be dominant allele, but the starch grains produced are intermediate size in baby seeds. So if a starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or product that it is information for. It is dependent as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine. In case more than one phenotype is influenced by the same gene. Inheritance of two genes. Mendel also worked with and crossed pea plants that different in two characters, as is seen in the cross between the pea plant that has seeds with yellow color and round shape and one that had seeds of green color and wrinkled shape. Mendel found that in seeds resulting from the crossing parents had yellow colored and round shaped seeds. Here you can tell which of the characters in the pair's yellow or green color, round and wrinkled shape was dominant. This yellow color was dominant over green, and round shape dominant over wrinkled. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use the genotypic symbols: Y for dominant yellow seed color and Y for recessive green seed color, R for round shaped seeds and R for wrinkled seed shape. Genotype parents can be then can be written as RRYY and RRYY. The cross between the two plants can be written down as in figure 4.7, showing the genotypes of parent plants. The gametes RY and RY unite on fertilization to produce F1 hybrid RRYY. When Mendel self-hybridized the F1 plants, he found that three-fourths of the F2 plants had yellow sheets and one-fourth had green. The yellow and green color segregated in a 3:1 ratio. Round and wrinkled sheet shape also segregated in the 3:1 ratio, just like in monohybrid cross. Law of independent assortment. In the dihybrid cross, the phenotypes round yellow, round green, and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as combination of series of 3 yellow, 1 green with 3 round, 1 wrinkled. This derivation can be written as follows: 3 round ratio 1 wrinkled, 3 yellow ratio 1 green, 9 round yellow ratio 3 wrinkled yellow, 3 round green, 1 wrinkled green. Based upon such observation, the on dihybrid crosses between plants differing into traits, Mendel proposed a second set of generalizations that we call Mendel's law of independent assortment. The laws state that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. The Punnett square can be effectively used to understand the independent segregation of two pairs of genes during meiosis and the production of eggs and pollen in the F1RRYY plant. Consider the segregation of one pair of R and R. 50% of the gametes have the gene R and other 50% have R. But now, besides each gamete have either R or R, it should also have the allele Y or Y. The important thing to remember that is the segregation of 50% R and the 50% R is independent from the segregation of 50% Y and 50% Y. Therefore, 50% of R bearing gametes has Y and the other 50% has Y. Similarly, 50% of the R bearing gametes has Y and the other 50% has Y. Thus, there are four genotypes of gametes. The four types are RY, 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 and RY, each with a frequency of 25% or 1/4 of total gametes produced. When you write down the four types of X or and plot it on the two sides of the Punnett square, it is very easy to derive the composition of zygote that give rise to the F2 plants. Although there are 16 squares, how many different types of genotypes and phenotypes are formed? Note them down in the format given. Now you can use the Punnett square data work out the genotypic ratio and at the F2 stage and the fill in the format given. It's the genotypic ratio also 9 ratio 3 ratio 3 ratio 1. Chromosomal theory of inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons that remained unrecognized in 1900. Firstly, communication was not easy as it is now in those days, and his work could not be widely publicized. Secondly, the concepts of genes or factors. Mendel's words are stable and discrete and units that control the expressive traits and their alleles which did not blend with each other, was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that the factor gene were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. In 1903, scientists D. Various occurrence and von Smark independently discovered Mendel's result on the inheritance of characters. Also, by this time, due to results in my advancements of microscopy and were taking place, scientists were able to carefully observe cell division. This led to discovery of structures in the nucleus that appear to double divide just before each division. These were called chromosomes, colored bodies as they were visualized by the staining. By 1900, the chromosome movement during meiosis had been worked out. Walter Sutton Theodore Boveri noted that behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's laws. Recall that you have studied the behavior of chromosome during mitosis, equational division, and during meiosis reduction division. The important things to remember are that chromosomes are as well as genes occur in pairs. The two alleles of the gene pair are located in the homologous sites on homologous chromosomes. During the anaphase of meiosis, first the two chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosome of four different colors in the left and right columns. And the left column are orange and green segregating together, but in the right hand column, the orange chromosome is segregating with the red chromosome. Sutton and Boveri argued that the pairing of and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principle and called it chromosomal theory of inheritance. Following this of ideas experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis of the for the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies, Drosophila melanogaster, which were found very suitable for just such studies. They could be grown on a simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there were a clear differentiation of sexes. The male and female flies are easily distinguishable. Also, it has many types of hereditary variation that can be seen with a low power microscope linkage and recombination. Morgan carried out several hybrid courses, Drosophila, and study genes that were sex linked. The crosses were similar the hybrid process carried out by Mendel in peas. For example, Morgan hybridized yellow body, white-eyed females to brown-bodied, red-eyed females to across their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from 9 ratio 3 ratio 3 ratio 1 ratio. Morgan and his group knew that the genes were located on the X chromosome, and so quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combination were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes, and they coined the term linkage to describe this physical association of genes on the chromosome, and the term recombination to describe the generation of non-parental gene combinations. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were tightly linked, showed very low count of recombination, while others were loosely linked, showed high recombination. For example, he found that the genes white and yellow were tightly linked and showed only 1.3% of recombination, while white and miniature wing showed 37.2% of recombination. His study Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of distance between genes and map their position of chromosome. Today, genetic maps are extensively used as a starting point in the sequence of whole genomes, as were done in the case of human genome sequencing project described later. Polygenic inheritance. Mendel's studies mainly describe those traits that have distinct culinary forms, such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not distinct, and their occurrence and are separate across a date. For example, in humans, we don't just have tall or short people as two distinct cognitive, but a whole range of possible heights. Such traits are generally controlled by the three or more genes, and others are called polygenic traits. Beside the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele. It means the effect of each allele is adaptive. To understand this better, let us assume that three genes ABC control skin color in human, with the dominant forms ABC responsible for the dark skin color and recessive forms ABC for the light skin color. The genotype with all dominant alleles AABBCC we all have darkest skin color, and that will the recessive alleles AABBCC will have lightest skin color. As equated, the genotype with three dominant alleles and three recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles and the genotype would determine the darkness or lightness of the skin and on an individual. Pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, substances where a single gene can exhibit multiple phenotypic expression. Where such a gene is called a pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the different effect of a gene on metabolic pathways which contribute towards different phenotypes. An example of this is the disease phenylketonuria, which occurs in humans. The disease caused by the mutation in gene that codes for a enzyme phenylalanine hydroxylase. This manifests itself through phenotypic expression characterized by the mental retardation and reduction in hair and skin pigmentation. Sex determination. The mechanism of sex determination has always been a puzzle for the geneticist. The initial clue about the genetic chromosomal mechanism of sex determination can be traced back although some experiment carried out in insects. In fact, the cytological observation made in a number of insects led to development of concept of genetic chromosomal basis of sex determination. Hanking could trace a specific nuclear structure all through spermatogenesis in few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas other 50% sperm did not receive it. Hanking gave name to this structure as X-body, but he could not explained its significance. Further investigations by the scientists led to the conclusion that the X body of Hanking was in fact a chromosome, and that is why it is given the name X chromosome. It was also observed that in a large number of insects of the mechanism of sex recombination is of exotype. It means all X bear an additional X chromosome beside the other chromosomal Autosomes, in the other hand, some of the sperms bear X chromosome, whereas some do not. X fertilized by sperm having X chromosome become females, and those fertilized by sperm that not have an X chromosome become males. Do you think the number of chromosomes in the male and female are equal? Due to the environment of X chromosome in the determination of sex, it was designated to be sex chromosome, and the rest of the chromosomes and named as autosomes. Grasshopper is an example of exotype of sex determination in which with the males have only one X chromosome besides the autosomes, where females have pair of X chromosomes. These observations led to the investigations of a number of species to understand the uh, sex mechanism of sex determination in number of others can the mammals including man. XY type of sex determination is seen where both female and female have same number of chromosomes among the males, and X chromosome is present but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosome. Both male and female bear same number of autosomes, and the males have autosome plus XY, while the female have autosome plus X in human beings. In the Drosophila, the males have one sex and Y chromosome, whereas have pair of X chromosome beside autosomes. In the above description, you have studied about two types of sex determining mechanism, exotype and XY type, but in both cases will produce two different type of gametes, either with or without X chromosome. Be some gametes with X chromosome and some with the Y chromosome. Such type of sex determination mechanism is regulated by the example of male heterogamety. Since some other organisms, example bird, a different mechanism of sex determination is observed. In this case, the total number of chromosomes in both male and female, but two different type of gametes in terms of sex. Chromosomes are produced by females, female heterogamety. The order to have In order to have distinction with the mechanism of sex determination described late earlier, the different sex chromosome of a female bird has been designated to be Z and W chromosomes. In these organisms, the female have one Z and one W chromosome, whereas the males have a pair of Z chromosome beside the autosomes. Sex determination in humans. It has already been mentioned that the determining mechanism in case of humans is XX type. Out of 33 pairs of chromosomes present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosome are present in the female, whereas the presence of X and Y chromosome are determinant of the male characteristic during spermatogenesis. About among during sperm among males, two types of gametes are produced, and 50% of the total sperm produced carry the X chromosomes, and the rest 50% have Y chromosome beside the autosome. Females, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case of the ovum fertilized with this carrying X chromosome, the zygote develops into the male, and the fertilization of ovum with Y chromosome carrying result into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determine the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or female child. It is unfortunate that in our society women are blamed for giving birth to female children and have been ostracized and ill-treated because of this false notion. So we will learn the rest of the chapter in the next. Sex determination in honeybee. The sex determination in honeybee is based on the number of sets of chromosomes an individual receives. An offspring formed from the union of sperm and the egg develops as a female, and then unfertilized egg develops as male by means of parthenogenesis. This means that the males have a half of the number of chromosomes than that of female. The female are diploid, having 32 chromosomes, and males are haploid, having chromosomes. This is called haplodiploid sex determination system of, and has special characteristic features such as male produces sperm by mitosis. They do not have father and thus cannot have sons, but have a grandfather that can have grandsons. How is sex determination mechanism different in birds? Is the sperm or egg responsible for the sex of chicks? Mutation is a phenomenon which results in the alteration of DNA sequence and consequently result in a change in the genotype and phenotype of an organism. In addition to recombination, mutation is another phenomenon that led to variation in DNA. As we will learn chapter 5, the DNA helix run continuously from one end to another end in each chromatid in a highly supercoiled form. Thus, therefore, loss or gain of a segment of DNA result in alteration in chromosomes. Since genes are known to be located on chromosome, alteration in chromosome result in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, mutation also arise due to change in single base pair of DNA. This is known as point mutation. A classical example of such mutation is sickle cell anemia. Deletion and insertion of base pair of DNA causes frame shift mutations. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induces mutation. These are referred to as mutagens. UV radiation can cause mutation in organisms. It is a mutagen. Genetic disorder. Pedigree analysis. The idea that disorders are inherited that has been prevailing in the human society since long. This was based on the inheritability of certain characteristic features in families. After the discovery of Mendel's work in principle of practice of analysis inheritance pattern of traits in human being began, since it is evident that control crosses that can be performed in plant or some other organisms are not possible in case of human being. The study of family history about the inheritance of a particular trait provides an alternative. Such analysis of traits in a several generations of family is called pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generation. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality, or disease. Some of the important standard symbols used in pedigree analysis have been shown in figure 4.13. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. DNA is the carrier of genetic information. It is hence transmitted from one generation to other without any change or alteration. However, changes that or alteration do take place occasionally. Such an alteration or change in genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with an change or altered genes or chromosomes. Mendelian disorders, broadly genetic disorder, may be grouped into two categories: Mendelian disorder and chromosomal disorders. Mendelian disorder are mainly determined by alteration or mutation in a single gene. These disorders are transmitted. These disorders are transmitted to the offspring on the same line as we have studied in the principle of inheritance. The pattern of inheritance of such Mendelian disorder can be traced in a family by pedigree analysis. Most common and prevalent Mendelian disorder are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. It is important to mention here that such Mendelian disorder may be dominant or recessive, but pedigree analysis one can easily understand whether a trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome, as in case of hemophilia. It is evident that this X-linked recessive trait shows the transmission from carrier female to male progeny. A representative pedigree is shown in figure 4.14. For dominant and recessive, discuss with your teacher and design pedigree for characters linked to both autosomes and sex chromosomes. Color blindness. It is a sex-linked recessive disorder due to defect in either red or green chromophile, resulting in failure to discriminate between red and green color. This defect is due to mutation in certain genes present in X chromosome. It occurs in about 8% of males and only about 0.4% in of females. This is because the genes that led to red green color blindness are on X chromosome. Males have only one X chromosome and females have two. A son of woman who carries the gene has a 50% of genes being color blind. The mother is not herself color blind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is carrier and her father is colorblind. Hemophilia, the six-linked recessive disease which shows its transmission from unaffected carrier female to some of the male progeny, has been widely studied in this disease. A single protein that is part of cascade of proteins involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in a non-stop bleeding. That heterozygous female carrier for hemophilia may transmit the disease of two sons. The possibility of female becoming a hemophilic is extremely rare because mother of such female has to be at least carrier of the father should be hemophilic. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was carrier of the disease. Sickle cell anemia. This is an autosome linked recessive trait that can be transmitted from parents to the offspring when both the partners are carrier for a gene. The disease is controlled by a single pair of alleles, HBS Out of the three possible genotypes of homozygous individuals or HBS who disease phenotype, heterogeneous HBA individual appear apparently unaffected by the carrier of the disease as the 50% of him probability of transmission of mutant gene to the progeny. The same sickle cell trait that effect is caused by the substitution of glutamic acid by valine at the sixth position of beta globin chain of the head hemoglobin molecule. The substitution of amino acid in the globin proteins result due to single base substitution at sixth codon of the beta globin gene from the GAC to GOG. The mutant hemoglobin molecule undergoes polymerization and under low oxygen tension, causing the change in the shape of RBC from biconcave disc to the elongated sickle-like structure. Phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine to tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivative accumulation of these in brain result in mental retardation. This is also secreted through urine because of its poor absorption by kidney. Thalassemia. This is also autosomal recessive blood disease transmitted from parent to the offspring when both the partners are an affected carrier for gene of heterozygous. The effect would be due to the either mutation or deletion of which such may result in the reduced rate of synthesis of one globin chains that make up hemoglobin. This causes the formation of abnormal hemoglobin molecules resulting in anemia, which is characteristic of disease molecule. The thalassemia can be declassified according to which chain of hemoglobin molecule is affected. In alpha thalassemia, production of alpha hemoglobin chain is affected, while in beta thalassemia, production of beta globin chain is affected. Alpha thalassemia is controlled by two closely linked genes, HBA first and HB8 second on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of four genes. The more genes are affected, the less the alpha globin molecules produced. While beta thalassemias are controlled by a single gene, HBV on chromosome 11 of each parent occurs due to mutation of one or both genes. Thalassemia differs from sickle cell anemia in that farmers is a contributive problem of synthesizing too few globin molecules. The latter is qualitative problem of synthesizing an incorrectly functioning globin. Chromosomal disorders. The chromosomal disorders, on the other hand, are caused due to absence of excess abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosome called aneuploidy. For example, Down syndrome results in the gain of extra copy of chromosome 21. Okay. 21. Similarly, Turner syndrome results in due to loss of X chromosome in human female. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosome in an organism, and this phenomenon is known as polyploidy. This condition often seen in plant. The total number of chromosome in a normal human cell is 46. Out of these, 22 pairs are autosomes and one pair of chromosomes are sex chromosomes. Sometimes, though rarely, either an additional copy of chromosome may be included in an individual, or an individual may lack one of any pair of chromosomes. This situation are known as trisomy or monosomy of chromosome respectively. Such a situation leads to very serious consequences in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome are common example of chromosomal disorders. Down syndrome, cause of this genetic disorder is the presence of an additional copy of the chromosome number 21. This disorder was first described by Langdon Don. The affected individual is short-statured with a small round head, furrowed tongue, and partially open mouth. Palm is broadened with characteristic palm crease. Physical, psychomotor, and mental development is retarded. Klinefelter syndrome. This genetic disorder is also caused due to the presence of an additional copy of X chromosome, resulting into a karyotype of 47XXY. Such an individual has overall masculine development. However, the feminine development of breast gynecomastia is also expressed. Such individuals are sterile. Turner syndrome. Such a disorder is caused due to the absence of one X chromosomes with 45 with X. So such females are sterile over as ovaries are rudimentary besides other features including lack of other secondary sexual characters. So our chapter is finished. Thanks for